Hello, everyone. My name is Sandy Vassos. I'm the Patient Community Director for the Fibrolamellar Cancer Foundation. I'm so happy to welcome you to the first in a series of virtual meetings scheduled for this fall, with more to come next year. Please visit our website, fibrofoundation.org, for more information. Now, throughout this event, feel free to submit your questions through Q&A, and we'll get to as many as we can by the end of the session. Now I'd like to kick us off by introducing our board co-chair, Marna Davis.
Hi, friends, newcomers. Good evening, good afternoon, good morning to some. We have people from so many time zones with us tonight. We've got 46 participants, 8 of you from out of the country. 13 of you have been to Vermont to our Fibrolamellar gathering, and I'm so glad you're here. And we also have 10 people from the Fibrolamellar Cancer Foundation, board members, staff, and I gotta tell you, this staff has been working really hard on this, our first of a series of webinars, which we will get better and better at, and I hope that you find it as helpful as we have found it. We've got fighters tonight. We've got parents. We've got siblings, spouses. We've got friends, survivors. We have those who have lost someone to fibrolamellar, but we are all part of the fibrolamellar community. We are a very diverse and wonderful lot of people, and we all come together for each other as one. I am Marna Davis, and I'm co-chair. My goodness, I didn't cry. I'm sorry, I didn't cry when I practiced this, but I am co-chair and proud to be that of the Fibrolamellar Cancer Foundation, and I am mother of Tucker Davis, who founded FCF with his friends in 2009, just before he passed away from fibrolamellar. And our mission is threefold, okay? It is number one, to raise money to fund research to find a cure and therapies for fibrolamellar. Number two, to raise awareness for this very rare disease. And number three, to support the fibrolamellar community, that's you, in your fight. And so addressing mission number 3 to support our fibrolamellar community. Welcome to virtual Vermont. What's that? What's virtual Vermont? Well, some of you have been to Vermont, and we hope that the rest of you will come. But we do something through the Fibrolamellar Cancer Foundation, which is really quite unique. And we've come to realize that it's really quite impactful for so many fibrolamellar fighters and their families. We hold an annual weekend gathering in Stowe, Vermont, beautiful Stowe, for fighters and their families and their friends, and for those who have lost someone to fibrolamellar. It's really an amazing few days, and it's really quite unique in this rare cancer world. And we take care of you. We take care of each other. It's a time to take a breath. It's a time to relax a bit. It's a time to share your concerns with others who get it. We're all in the same boat. And it's almost hard to put into words what it means to so many of us. You have to come and see for yourself, okay? And there's not a big agenda for this weekend, although we have a lot of wonderfulness built into it. And that said, on the Saturday morning of this weekend in September, we gather after breakfast, and the Fibrolamellar Foundation gives an update on what we've been doing that year, as well as a state of the art on fibrolamellar research. But know that there's a lot of doing that's needed to support the all-important research, okay? So it's not just research. There's so much that goes into running this foundation, and we're so busy all year long in getting all that done to support the all-important research that we are funding. So let's pretend, everyone, that we're all sitting in the fly room of the Lake Mansfield Trout Club. The leaves are beginning to turn. The breeze is blowing in off the lake. We have a hot cup of coffee in our hands. There's a fire in the fireplace. And here's John Hopper, our president, Mark Furth, our scientific director, about to fill us in what's going on in the fibrolamellar cancer world. Thank you. Here's John.
Thank you so much, Marna. And I had to put this picture up, Marna, as the way you described the Vermont outing, because this was the last one we had in 2019. I still have my hat from the first one I went on. You can see it's been worn quite a bit. But you can see the partnership, the collaboration, just the, quite frankly, the love in the group that we have. And I needed to share this before I started the talk about what's been going on with the foundation. And what it is that we've been building momentum through collaboration and partnership. So again, I hope everyone who's on this Zoom who hasn't been to Vermont, like Marna said, please join us next year. So what we're going to do today is I'm going to introduce you to the folks at the FCF. I'm going to talk about some new partners and collaborators that are really helping us accelerate research. Dr. Mark Furth is going to come in and talk about our research top line, and then we're going to have time for Q&A. So who are we? Well, you just met Marna. You just met myself, John Hopper. Meet Chuck Davis, who is Tucker's dad and our co-chairman. We have a fantastic board of directors of 11 people that goes from everything from 3 of the co-founders with Tucker to the person who actually named Fibrolamellar, to someone who's at the Duke Cancer Institute, to marketing executives. It's really a top-notch, passionate group of board directors. Meet Dr. Mark Furth, our scientific director, who'll be talking momentarily, Ann Adler, who's a board director, but also oversees our social media. Lynn O'Malley, who many of you know, who's been with the foundation since the start, who's our community manager, and Megan McKinney, who's kind of our engine and doing all of our administrative work, and we thank them all. I also want to introduce you to an expanded team. As the needs of the community continue to grow, so do we. You just met Sandy Vassos, our patient community director. Manny McGowan, one of our fighters, has come on as a consultant, as a patient resource manager. I'll get into that in a minute. Kurt Losart, one of our caregivers, has been doing a fantastic job really with all of our digital and social media. Patty Cogswell, some of you met, is actually running our biobank. And Jenny Carroll, also one of our caregivers, has been a great creative consultant. Again, we welcome them all helping us this year. So, big news for 2020. Fibrolamellar Cancer Foundation is the inaugural recipient of the Chan Zuckerberg Initiative Rare as One Network Grant. So for those of you who have never heard of the Chan Zuckerberg Initiative, look them up. They are now what I call the modern day Gates Foundation, Rockefeller Foundation, a multi-billion-dollar foundation that actually has a love for rare diseases. So they've created this thing called the Rare as One Network. And you can see Mark Zuckerberg, the founder of Facebook, and his wife, Priscilla Chan, with a motto behind it that says, we believe we can cure all diseases in our children's lifetime, but with a particular emphasis on rare diseases. So what does all this mean for all of you and all of us? Well, the Rare as One Network has a mantra, and this comes from Mark and Priscilla. They believe that a proactive patient research community will help accelerate discoveries. What does that mean? It means patients and researchers working together to find a cure, not just the researchers, but all of us and all of you. Their approach is very simple. They actually went out and asked for applications, got 500 applications from rare disease organizations. They picked 30 who they thought had the highest potential to make progress in their diseases. What are they doing for us? They're creating a shared learning network amongst all 30 of these rare diseases. We're one of 3 cancers, by the way, in there. They're offering expert consultative access from some of the top leaders in the country. They're funding research and infrastructure. We have almost $500,000 over the next 2 years from the Chan Zuckerberg Initiative. And it's a 2-year commitment. This is not just an in and out. They believe they can help us make progress. And God bless them, we are making progress. What else is new? If any of you have looked to donate to an organization, you probably have gone to something called a Charity Navigator. It's the place to look for foundations. Say, how are they being run, and is it worth you donating? Well, we just got our first score is 100 out of 100, which basically means give with confidence. And that's because of the stewardship and the integrity of the foundation. When we get money in, you all should remember that all donations go to research. The Charles and Marna Davis Foundation covers all of our operating expenses, an important aspect to note, but we are truly proud of this. And not many foundations, large or small, get 100 out of 100. On that, over the 10, actually now 11 years we've been in existence, we have committed over $8 million in research across 24 institutions. I'm not going to read all this. I'll keep it up for just a second, but you can see all the fantastic organizations that we have funded, many of them active, to find a cure for fibrolamellar, internationally and nationally, across the board, pediatric institutions, as well as other major academic institutions. We're very grateful that we have this group working with us. So along that line, in 2019, we had our third biennial Fibrolamellar Cancer Scientific Summit. So that brought together over 30 different institutions, over 80 researchers, 5 pharmaceutical companies, biotech companies, the National Cancer Institute, all together. But why do we do that? Is to create a roadmap for the future. How do we want to spend our research dollars? What's the emphasis? What are the important points that are going to help make a cure? And we hear over 2.5 days from the experts in the field, from Rush University to MD Anderson, to the Mayo Clinic, to Johns Hopkins, you name it. We hear from them as to what they believe will move things forward for fibrolamellar, and it gives us that roadmap so we know, not just the people who are managing the foundation, but outside, what's the best way to spend our investment dollars and our efforts, and hopefully have great results. There we go. Okay, so Kurt Loser, I told you, is come on board, and he is magical. He has actually enhanced our website. I would ask everybody, please go to the website and take a look at it. What we need here is communications that's dynamic. So if you're newly diagnosed, you want to know about fibrolamellar, you want to know what's going on with research, you want to know what patient researchers are, clinical trials. We've got it all. You want to join the organization and do things, join the fight. But please take a look at this newly revamped website. And again, we will constantly be adding news to this. So if you saw it today, there's going to be news again next week. Thank you, Kurt, for your efforts on this. Okay, other partnerships that we have in collaborators. Pharma and biotech companies, as I mentioned before, have taken an interest in fibrolamellar. Bristol Myers Squibb, Alnylam, Puma, Genentech. In fact, we get our first corporate grant from BMS just recently. Venture philanthropy basically is people with a lot of money who want to give to good causes. We discovered the Mark Foundation for Cancer Research, and because of our relationship there, we were able to get St. Jude's Children's Research Hospital $250,000 for 2 years of research on fibrolamellar. Many of you have actually helped to review Department of Defense reviews. Well, you know what? It's working out. Over $2 million the Department of Defense has funded for fibrolamellar research, and it's active research right now. And the NIH, National Cancer Institute, looks at the fibrolamellar audience and says, we want you to be part of our My Part initiative, which is my pediatric and adult rare tumor network. So we are one of the featured cancers. They are looking to do all types of research and advance the progress for us. We're very fortunate. Advocacy also comes into collaboration. There's a lot of different groups working on fibrolamellar and other cancers, but we want to work together. We're very blessed that Tom Stockwell with his Fibro Fighters continues to work with us, and together we are looking at, advancing biospecimen collections, looking to create something new. Stay tuned, a Fibro Forum launch and other activities. Thank you, Tom, for all your collaboration. And we also look at other major associations like the Cancer Research Institute, which actually was the first group to think of immunotherapy, I think over 50, 60 years ago. And we actually fund fellows through them. ASCO, which is the largest oncology organization. NORD, the largest rare disease organization, GI Cancer Alliance, and many, many others. Along that fact, NORD, along with Fibrolamellar, created the Rare Cancer Day. Last year was the first year. This is the second year. And we're going to be featured because the U.S. Senate and Senator Richard Blumenthal recognizes Fibrolamellar. I just got this hot off the press last night, so I'm going to read what's going to be in a press release next week. So bear with me. This is from Senator Blumenthal. On Rare Cancer Day, I'm proud to stand with the Fibrolamellar Cancer Foundation and the National Organization of Rare Disorders to raise awareness of rare, often deadly cancers and to keep up the fight for cures. With robust federal investment, we can and will find cure for rare cancers like fibrolamellar and aggressive cancer impacting our youth with no effective cure. Increasing ongoing federal funding is essential to spur innovation and discovery of treatments and cures to help save lives. Senators like Blumenthal have been a friend of fibrolamellar for many years, and we're going to keep him to his word. So everybody, on the 30th, come celebrate Rare Cancer Day with us all. Last but not least, what do I want to leave with you? The thought is that the fight against fibrolamellar depends on all of us and all of you. Please get involved. How can you get involved? You'll be hearing about our newly launched biobank. We need tissue donations for researchers to do their job. Information sharing, I mentioned the fact that Kurt and Tom and others are looking at forums for us to share information, share it on our website, share it on the registry, but share your information. Look for participation in research. There's clinical trials out there. Ask your oncologist, ask Mark Furth about those. Are they appropriate? What do you need to know about that? We're also going to launch something about Meet the Researchers over the next year or so, where you can meet some of the researchers doing the study so they understand your journey. They'll learn more about where the research should go. And also just help build awareness locally, state, nationally, in any form that you can do that. Your engagement provides the most important key to success, which will help lead to a cure. So before I pass this over to Mark Furth, I'm going to use a term that Tucker Davis came up with when he found the foundation, which is let's all keep on tucking. Mark, over to you.
Thank you, John. You know, we do miss Vermont. I first went to Vermont with John and Marna and Chuck and the whole crew 2 years ago, totally wet behind my ears, just about to start as the scientific director for this foundation. And an overwhelming memory from that time was sitting down by the fireplace with some of the younger patients who were there and talking really deep into the night, 2, 3 in the morning, about their stories. And at the time, and even still today, most of you who have dealt with this cancer know that we're still dealing with the old-fashioned triad of treatment options that have been around for cancer for a very long time. If you're fortunate enough that your tumor has not spread badly, even if it's a large liver tumor, you're initially going to undoubtedly seek out surgery, maybe in a very extreme case, a liver transplant, but more likely resection. You may have some radiation as part of your procedure. And depending on your situation, especially if the cancer has evidence of having spread outside the liver, you'll have some form of chemotherapy. You know, if you have a couple of big lung mets, maybe the surgeon will take them out, but you're certainly not going to be cured of that kind of disease by what we have today. There's no standard of care. There are few cases with proven long-term benefit from chemotherapy or any other therapy other than surgery. Not a great picture. And yet we're here talking about maybe there's a possibility to use the word cure. And some of this is really because there's a modern world of cancer therapy, which is emerging and maturing and becoming really the standard of how we try to do research in cancer. And the terms that are going to be used tonight and you'll hear over and over are targeted therapy on the one hand and immunotherapy on the other. Today, I would say that the vast majority of our scientific roadmap for how we're going to try to get to curative therapies for this cancer within your lifetimes surround those two concepts. So I'm going to talk about them, and I'll try to stay away from a lot of pure lab or science speak and try to, as Marna always asks me to do, speak English. And if I fail at that, I hope the questions will come fast and furious at the end. But let's talk about what targeted therapy is. A lot of targeted therapy recognizes that cancer is a genetic disease and that mutations in certain proteins create altered proteins that can drive the cancer. Other mutations occur but have less of an effect, and we sometimes call those passengers. So this is the driver as in the driver of a bus. It's what makes that cancer continue to grow out of control and create the havoc that we're ultimately wanting to stop. So one of the key areas is to understand the kinds of growth signals that are involved in driving cancers, and specifically fibrolamellar, of course, and what are the biochemical pathways that maybe one can hope to interfere with in order to put up a stop sign to that growth, or maybe even more than a stop sign to get the cancer to commit suicide, if you will, to die. Another part of targeted therapy has been understanding more about the metabolism of cancer in the context of how it differs from normal tissue and whether that's something that can be leveraged for better therapy. Another area that's somewhat related to that is trying to choke off the blood supply to cancer, because cancers, of course, need blood supply, particularly when they're rapidly growing. So you can sort of take almost a humorous view of it, do not feed the wildlife, but seriously, this is a new part of the armamentarium, which is different than old-fashioned chemotherapy, which is essentially throwing toxins at cells and hoping that they're just that little bit more toxic to the rapidly growing cancer than to your normal tissue. An extreme version, and one that I'm actually quite excited about in cancer, what you might call molecular smart bombs. And that's really finding better and better ways to use what might in fact be very old-fashioned kinds of toxins, but to direct them extremely precisely only to the cancer cells so that you don't get the side effects that you see today. And we're moving to a generation where one of the tools for delivering those are antibodies. There are other ways to deliver them, which can recognize very precisely some aspects of the cancer cell surface that are different from a normal cell. We're also in a position where the nature of those payloads, the toxic compounds, and the way they're linked to the delivery systems are getting better and better. So these are the kinds of areas we'd like to develop rather than the more traditional kind of chemotherapy. And there's nothing new or special about this. This is where the whole field is going. The second major modality, which has really come into its own in the last few years, although as John said, it's something that people have been dreaming about for decades, is immunotherapy. And really the goal here is to enable the patient's own immune system to do something that's been evolved to do and programmed to do for millennia, or for as long as we've had life on this planet of a multicellular variety, which is to find renegade or rogue cells and eliminate them before they damage the whole organism. Cancer's been with us on the planet for a long time, and the immune system's pretty good at smelling it out and stopping it from growing. Unfortunately, cancers also evolve in many cases to bypass the surveillance of the immune system. Cancers can put up a kind of defensive shield, taking advantage of what are technically known as immune checkpoints. And there's been a new generation that many of you have heard about of what are called immune checkpoint inhibitor drugs. I'll probably just call them checkpoint drugs at most of the talk right now. And I think of those as arrows that can pierce that shield, or if you want to get a little more fanciful, sometimes those of you who are still Star Trek fans think about a cloaking device which protects the cancer, and these drugs are a decloaking device so that you can see it if you're an immune cell and go after and try to kill it. A second class of therapy that is related is to, in some cases, see if one can actually program a patient's immune cells to do a better job of attacking the cancer. And there are, in fact, a class of cells that we call T cell killers or killer T cells that are exquisitely specific in targeting certain classes of cells that the body needs to get rid of. They might be virus-infected cells, they might be cancer cells. Can we make them do a better job? Can we use genetic engineering or some other aspects of biology to increase the number of these cells, increase their specificity, and protect them from being blocked by the shields that the cancers can put up. So these are areas we really want to think about hard. Another area, and I won't dwell on it today, although we'll talk about this in the context of the biobank, that is crucial is not only to develop the therapies, but really to have the infrastructure that allows one to do the research biology and the drug discovery in a way that will get us to the goals for fibrolamellar itself. As is true for many rare cancers, we don't have some of the same laboratory resources for fibrolamellar that people would have for colon cancer or lung cancer, where different kinds of laboratory models have been developed for decades. So we need to be able to do a better job of growing human fibrolamellar cancer cells in laboratory culture. That starts with having the actual cells from patient biopsies available, and that's where the biobank comes in. We also, as the field has learned how to do for some decades, sometimes grow those tumor cells as tumors in mice that don't have good immune systems. And so these are sometimes called PDX models. You may have heard that term. Those two ways of working with the actual human cancer cells are crucial to making progress. Another thing that we'll do is use animals not to grow human cells, but to mimic them. One can put into the mice, for example, the same kind of driver gene that causes fibrolamellar or causes any other cancer, and that cancer driver gene can, in fact, generate a tumor that in many cases mimics the human cancer pretty well and allows one to do a variety of experiments that in some cases you can't even do with the human tumor cells, like look at a cancer in an animal that has its own intact immune system. So in fact, that tool was critical in work that was, I think many of you know, really sort of seminal in the field and allowed the real big intellectual breakthrough and practical breakthrough that's fueled so much of the research roadmap in fibrolamellar for the past 6 years. The Simon Laboratory at Rockefeller University, at the time funded by the foundation, made an important discovery that in a set of fibrolamellar patients, every one in that individual's tumor had a very specific kind of mutation, and one that's found in other cancers, but a little unusual relative to many cancers. It turned out to be a loss of DNA, a deletion, as we would call it technically, that brought two genes that are otherwise separate and unrelated very much closer together in such a way that it actually created a hybrid or chimeric gene, or fused gene. These terms are all synonyms. Shown here in this little picture with yellow for the part that comes from a gene that codes for a protein called protein kinase A, and another one that codes for a heat shock protein with the funny name DNAJ. But we'll just call that the chimera tonight. And although, technically you really should probably have a centaur, not a three-headed or a two-headed beast with a third organism's tail, because we've only got two components. The chimera, however you name it and however you think about it, opens the door to targeted therapy of fibrolamellar cancer, because in the pure form of the disease, it looks like maybe as many as 99% of patients share exactly this driver. And that's something that's really unusual in cancers. And it gives us the opportunity to really focus on understanding the signaling and the pathways that we might want to go after. And if we can generate a cure, it's one that could apply to a very large fraction of the patient population. An important discovery over the last couple of years, and I'm going to try to give you a couple of highlights that I think are important towards our progress, was from the laboratory of John Scott in Seattle, Washington, at the University of Washington. Other labs are working on similar things, and there's been related contribution recently from a terrific lab in San Diego. But the understanding is that one has to think not only about this driver protein itself, which is shown here in a little green circle in a cartoon where the pKa part, the main part with sort of the big activity, the protein is in blue, and this little extra head from the other gene is shown in yellow. So that protein is actually part of a bigger complex, and it interacts with proteins that are normally there as part of growth control and kind of a signal exchange or a relay for information in the cell that, among many other activities, is really pivotal in whether the cell is going to actively grow or not. And if something goes wrong here, there's the potential the cell will grow when it's not supposed to. One of the discoveries that the Scott lab has made is that because this is a chimeric protein and has this little extra domain, it qualitatively behaves in a different way from the normal pKa, which is a very well-known enzyme. In fact, John worked for some years with the discoverer of that enzyme and holds a chair at the University of Washington named after his mentor, who was a Nobel laureate. But we now know that pKa itself, even if it's made at higher levels than normal, does not transform a cell and create fibrolamellar cancer in that mouse model we just talked about. The fusion protein, the chimera, qualitatively is different. It has a much more potent activity in causing cancer. Why is that? One possibility is that it brings another protein into the complex that's not normally there. And that may change the interaction with a bunch of other proteins, and don't worry about what their names are, but these are well-known proteins involved in growth signaling. And there's something about this aberrant complex which makes these more active and less easy to turn off in giving growth signals so that they drive the growth of liver cancer cells. One of the thoughts that John has published with his group is that it may be that actually targeting a combination of proteins in that overall complex may be more effective than going after any one. Obviously, the most ideal thing would be if you could actually knock out the chimera itself, but there are technical reasons that that may not be so easy without causing damage to the body, because the protein, the blue part here, is also really critical in other functions like how the heart works, how insulin works, and so on. So we don't know if we can go after that, but maybe we can find novel combinations that will work on these aberrant complexes. Another approach that's been taken, and this has been led by Praveen Sethupathi of Cornell University, who's been working on fibrolamellar now for about a decade, is to ask, can we just identify genes that the fibrolamellar cancer needs to survive or to grow? And this is part of a much bigger picture. We're, for example, collaborating with the Broad Institute of MIT and Harvard on looking at that question systematically. Praveen has used tools that geneticists have developed to understand what genes are just locked on in the cancer by things in the chromatin, or the controls how genes are used, that have the great name of super enhancers. So they're locked on and are turning out certain proteins in a way that you might say the cancer is addicted to. It needs those. And among the set of genes he's identified that have that property of really being in that constant on and driven state in fibrolamellar are some attractive new candidates that might be targets for therapy, some of which you might never have thought about in other contexts. So that's yet another approach that, in fact, again, one would like to take even more broadly. So just to summarize where we are with this approach to targeted therapy, again, an ideal thing would be if we could find a way to either selectively block the activity or maybe the production of the chimera. And in fact, we're embarking in some collaborative work through the Cornell group and a company, Alnylam, on one strategy that might allow us to block the production of that fusion protein. There are others that we have at least outlined on blackboards and are hoping to find people to work on over the next year or two as part of an expanding roadmap in that area. As we already said, inhibiting downstream steps that control cell growth pathways that are ultimately driven by the chimera is another possibility. If you could find targets that either already have existing drugs in development or combinations of those, perhaps with one existing drug, something still waiting to be developed and so forth. But these possibilities of combinatorial approaches based on better understanding of the biology are very attractive. And again, we said you could target genes that are locked on by super enhancers and also to use the models that we've talked about to enable the largest possible scale of screens for targets that are necessary for survival of the cells and drugs from large sets that just really, every compound that's ever been shown to be safe in a human being, which of those might be beneficial against fibrolamellar. And in fact, that's part of our long-term collaboration with the Broad Institute. I want to turn now to immunotherapy. And I'd like to start with just what I think is a terrific graphic view of, nice if I could get the movie to play. Let's go again. I wanted to show you what happens when the right kind of T cell finds a cancer cell. Maybe I'm going to have to send you to this another time. Sorry about that. There we go. So green here is a killer T cell. The blue cell in this video is a cancer cell. The two little red dots actually show when that T cell is lining up to attack the cancer cell. It's a sign that it's found the cell. So you see those two dots get in position. That T cell is now about to start the process of zapping the tumor cell. Wouldn't it be great if we could find an army of those T cells in patients and get them to do that to the cancer cells? This is the same thing, but just with different filtering. So now that T cell appears bright golden, and you get a real idea of how intimately it's wrapped around the tumor cell as it's undergoing the very specific process of destroying it. But as we said, sometimes you can have a T cell, shown here in the bottom left panel as the blue cell with a little sort of purpley color nucleus, going after a tumor cell. And in this case, in fact, that T cell should be able to kill the cancer cell because it's got the right lock and key structure to find something that's specific about the tumor cell that is the trigger for it to want to kill it. We call that a receptor and an antigen. Don't worry about the language again. But in this particular case, something else is going on. One of these so-called immune checkpoints is intervening something that the tumor cell is matching a different surface receptor on the T cell. And it's saying, don't kill me. You're not supposed to kill me. It's overriding the normal behavior of that killer cell and putting up a shield. What the checkpoint drugs do is basically unshield the tumor cell and do that by destroying the interaction between that second signal. It gets rid of the don't kill me signal so that in fact the T cell does its job of killing the cell it should be going after. And that leads to the question, can immune T cells in fact kill fibrolamellar cancer cells? There's some reasons to think that fibrolamellar cancer cells might be what people sometimes call immunologically a little bit cold. That is, not really good candidates to be killed by the immune system. But that may not be true, and we're going to try to find out when T cells can actually kill the fibrolamellar cancer cells. And we want to ask very specifically, do FLC cells use the immune checkpoint system to evade? Do they put up the shields? And can those shields be overcome by inhibitor drugs? And just the names of some of them, which many of you may be familiar with, are shown on the slide. What we've heard so far, although nobody's done a very systematic study, there's in fact one in place at the Dana-Farber Cancer Center at the moment, but it appears that single checkpoint drugs don't do a very effective job against fibrolamellar. And that may not be so surprising, given that they're not in that kind of hot level where they have so many mutations that the immune system can find them easily. So the question is, can there be ways to enhance that? There's a clinical trial which is coming up that we're funding at MD Anderson Cancer Center that actually builds on some work that's already being done at Rush in asking the question of whether a combination of two drugs that have been used for some years in fibrolamellar, many of you may have even had those drugs yourselves if you're patients, would be a combination of 5-fluorouracil and interferon, but now asking, do those drugs potentially synergize with a checkpoint drug to kill fibrolamellar? And that's an approach that, again, mimics what's going on elsewhere in the cancer research field. Another thing which has happened in recent time is that people have begun more and more finding ways to be able to use more than one of these checkpoint drugs at the same time, which work against different shields, two different shields. And there's at least one very encouraging case report that appeared early in 2020 from a laboratory and hospital in Germany saying that one very fortunate young woman who had a very advanced case of fibrolamellar, a combination of two drugs, in fact, gave her a really strong response, and her tumor is essentially virtually gone. And I just spoke to Dr. Dottoni a couple of weeks ago, and the patient is still doing extremely well. So this dream of creating an army of anti-fibrolamellar T cells is a real one. And you can imagine kind of a Clone Wars scenario where you'd like to have not one lonely fighter with the gun that's specific for fibrolamellar, but turn that into an army. Well, how do you expand them to create an army? If you can find one, is there a way to go from one to many millions? And the answer is there are some ways. One is to use genetic engineering of T cells to create the army, sort of manufacture it, and then put it into play. But another thought is one that was brought to us by a young physician scientist from Johns Hopkins named Mark Furth. He came up to John Hopper and me at a ASCO meeting in January of 2019 and said, hey, maybe we could vaccinate patients, not to prevent disease because they already have it, but maybe we could create a therapeutic vaccine for fibrolamellar that would expand any T cells they have that are capable of killing fibrolamellar. And the very specific question he asked, and one which I thought was really logical, and I've actually been asking it since about the day that I joined the foundation, is can the chimera protein be a target for the immune system? Well, why is that an interesting idea? Because the driver of a cancer, if it becomes also the target for an immune system, gives the immune system and those who want to treat the cancer a great advantage. It's hard for the cancer to lose that driver. Usually with immune therapy, you worry, if it's just sort of an accident, the protein has a signal for the immune system to find. Maybe you can throw away that signal and still grow as a cancer. But if it's the driver gene that's being attacked, it's not so easy, and especially for a cancer like fibrolamellar that's a little more stable than some other cancers and is probably locked into the same driver much more tightly. Some cancers can generate new drivers really fast. Fibrolamellar doesn't seem to do that. So here was the idea. The green part of the protein is normal. The blue part of the protein is normal. The immune system won't react to those. The junction between the two is not there in normal cells. Maybe the immune system can pick out that junction. So what Mark Furth said, all right, well, let's try to vaccinate with a little piece of the protein that corresponds to that junction region and see if we can't get patients to amplify an immune response that way. And so that clinical trial actually began with our support and also support of Bristol Myers Squibb in March of this year. And I'm saying that was quite heroic, both from the patient and the physician point of view, because it occurred right in the middle of the COVID crisis in Maryland. Yet Mark was able to start the trial in which he's going to try this vaccine plus the two checkpoint drugs, the same ones that were used in the German patient, and ask two questions. Can patients develop immunity against the cancer? And does that give clinical benefit? So we're following that with great excitement and sort of bated breath to see what happens. So let me just summarize what I've been saying. Is there hope on the horizon? I'm convinced, yes, there is. I think there's some rational approaches in targeted therapy. The idea of the chimera protein as a drug target, these signaling islands, or finding genes that the cancer depends on, or just a marker that we can use as a way to bring a smart bomb into play to kill the cancer. On the immune therapy side, the combination of understanding how and when to use checkpoint drugs in combinations that may be more effective in this cancer, targeting the chimera for its attack, finding other possible natural targets for fibrolamellar for the immune system, or using T cells as the payload in spark bombs. And there's a kind of therapy called CAR T where you could do that. So I think there are a whole range of possibilities. My goal as scientific director is really to just take a lot of rational shots on goal against this cancer, with the belief that the technology is now moving to the point where a lot of that can be done. In that process, we have a marvelous network of scientists and physicians dedicated to work on this cancer. And we've formally, in the last year, actually asked 6 of them, partly through the CZI initiative, to join our scientific advisory board. First time the foundation's actually formalized that. Their names are here. They include some of the leaders in oncology, immunotherapy, targeted therapy, and cancer genetics. And they're the people who keep me informed of what we should do, and really doing it all for the benefit of the heroes, for you, the patients and their families, who, with your courage, make all of this research possible. With that, let me pass the baton over to my colleague, Patty Cogswell, and she'll tell you a little bit specifically about the biobank and why we need your help in filling that bank up.
Thanks, Mark. First, I want to apologize that you all can't see me. I'm having some internet problems, so I'm just talking on a regular landline. I'm just going to give you just talk for a few minutes about the biobank. I just wanted to first say, we've mentioned the biobank several times. What actually is a biobank? Some of you might be wondering. It's a collection of tissue and fluids that are stored securely. In our case, our biobank is housed at MGH. And as Mark's been mentioning, they're used to create models of the disease. We're desperately needing models of fibrolamellar, and also to get tissue to researchers. So my biggest point that I'd like to make is that patients are the center of this. Without your tissue, there would be no biobank. You are an integral part of this research initiative, and now that we have the process set up, I'm hoping that anyone who's needing surgery or has a fluid draw will contact us, and we can work together to do this. It's important to know that in relationship to this tissue donation, there's no cost to you. The foundation covers that cost. It doesn't affect your care. There's no difference in how you're treated at your doctor, with your doctor. Your privacy is protected. No one will ever know what tissue is yours. We call that de-identified. And finally, and I think most importantly, this excess tissue that you would be able to donate is generally discarded, wasting this precious resource. It's something that's it's like gold. It's a commodity that we really need to be sure we're making the best use of. So my goal so far in the foundation, and as I continue, is to streamline the process, making it simple for patients and for families. The way this would work is if you were having surgery or a fluid draw, you would contact me, and I'll work with you to get assigned consent. Once we have that, you're done. I take over. I do the rest of the work to ensure that the tissue gets where it needs to. We work very hard to get tissue to all the researchers who need fresh tissue, and to store tissue in the bank for later use. And this is not, as Mark's mentioned, we started this about a year ago. This is not the future. We are operational now. We have had great success in our first year, despite COVID, where many of the surgeons, I'm sure most of you know that most of the surgery shut down during that time frame. We have cell lines in development and xenografts in development. We're distributing tissue to researchers, and we're really building a large number of samples that we can work with over the following years. I'll end by saying thank you to the patients for working with us to advance research. I had one patient say to me, and it's really stayed with me, that they didn't get to choose having cancer, but they do get to choose the ability to fight it potentially through donating tumor tissue. They felt empowered by doing this. I was moved by that, and I remember that whenever I'm talking with a patient. Together, I think we can find better treatments and hopefully a cure for FLC. With that, let's go back to Marna. Thanks, everybody.
Thanks, Patty. When Patty first told me what she was going to say, I just said, I really wanted to add something, because I remember back 10 years ago when Tucker had his 2 surgeries, and then later he had the fluid draw, you know, the ascites drained from his abdomen. Oh my gosh, there was no Patty Cogswell helping to do any of this. And Tucker's friend Alicia and we spent so much effort trying to pull together paperwork to deal with the surgeons, because they were so specific about what they wanted, and to get that tissue and that ascites to where it had to go. And it, oh my gosh, we didn't know what we were doing. And it really took so much effort. And since that time at the foundation, we've spent a lot of time trying to get a biobank going, and where is the best place to house it. And we have collected tissue, and the tissue has come in and you know, bit by bit, and it's been great. I mean, those of you who have donated tissue, it's been great. But I have been told by Nabil Bardisi at Harvard at Mass General, that's what MGH is, Mass General Hospital, and it's that the tissue now, the tissue collection process is humming. It is running like clockwork. So Patty takes over. Once you've talked to her, she contacts the hospital, the surgeons. She's got the paperwork figured out. I mean, she handles it. Oh my God, I feel, I get chills. I feel how so much simpler it is for patients and their families to do this, because this tissue is so important. Without that tissue, those researchers have nothing to research. They have nothing to test their theories on. So, and with a rare cancer like fibrolamellar, we need a lot of tissue, because they need it from many patients to be able to compare one to another, to another, to another. So it is so important that we get it. We're there now. It's taken us 10 years, but we finally got there. And again, I thank our team near and far, and thank our patients and their families for, you know, sticking by and sticking with us to get it down pat. And it is down pat. And thank you everybody for doing that. So it's great. It's great. It's a good thing. Okay. Am I supposed to pass this along to somebody?
I'll take it from here, Marna.
Thank you.
It's Sandra Vassos. Thank you so much, Marna. We definitely have lots of questions that have come through. So I'm just going to dive right into them from our audience. And the first one that really, the first one is directed to John. Is there going to be a Vermont 2021?
Great question. The answer is, God willing, yes. We have all the plans to do that. Lynn is on top of it already, making reservations at the Mount Mansfield Trout Club. So mark it down. We'll be announcing that as soon as we have some dates there. But typically, for those who haven't gone, it's usually the end of August to mid-September, in that time range. So we hope that there will be. In the meantime, though, as Sandy was saying, we have sessions coming up next week, the week after, set up for patients to meet other patients. We have one that's for 18 and over. We have one for 17 and under. And we have one for caregivers. So the great camaraderie we had, and we continue to have with our community, but those who want to be able to meet each other and speak to each other, we're going to be doing that through the Zoom technology. So we're going to try our best to get that happening. And it will start happening next week. So, but keep in mind, next August, September, dates will come up, and Lynn will give us more details on that and post that as soon as she has information.
Great. Thank you, John. The next one that I have here, I'm going to toss to Mark. Has there been any more research on getting a vaccine for fibrolamellar cancer?
Thanks, Sandy. I actually was describing the work on that, and by a vaccine here, we mean a therapeutic vaccine, something that could help a patient who already has the cancer. We haven't, as a field, figured out how to do prophylactic vaccines against cancer. But indeed, the clinical study that is now underway at Johns Hopkins is precisely on that. Liz Jaffe, who is a member of our advisory board and the mentor for Dr. Yarchuan, is one of the world leaders in developing cancer vaccines. She was actually also the president of the American Association for Cancer Research a year back, an extremely well respected and connected leader in immune therapy for cancer. And she's been trying to make therapeutic vaccines for a whole range of cancers. Fibrolamellar was actually attracted to that group because of sort of the genetic purity of it and the possibility of targeting a fusion protein. So we're following that very closely. And I know that Dr. Yarchuan is already beginning to get data on whether an immune response is being mounted in some of these patients. So that's certainly a very key area of our roadmap at the moment.
Thank you, Mark. Another one for you, Mark. When you were talking about T cells and immunotherapy, the question is, do those therapeutics have equal response rates for diseases at higher stage?
It's hard to, there's not enough known to really say quantitatively, but what one can say is that what's been remarkable about immune therapy is that there are a number of cases of patients with advanced stage disease who are showing good responses. One of the famous ones in the United States is Jimmy Carter, former president of the United States, who had an advanced cancer, I think it was a melanoma, that responded well to immune checkpoint therapy. And the example of this young woman in Germany, she had advanced stage 4 fibrolamellar, had failed chemotherapy, and responded to the 2 checkpoint inhibitors with near complete response. So there is the potential for even some advanced stage patients with the right immune therapy to get big responses that last a long time.
Thank you, Mark. Now we have we have 1 and 2 perhaps for Patty. 1, I'm going to read this verbatim, Patty. I quote, "I didn't know of the FCF biobank prior to my surgery, and my surgeon hospital sent my tissue to where they would any. Is it worth me trying to find out where exactly my tissue was sent and have it sent to the FCF biobank?"
Thanks for that question. It is something that we could do. It depends on the way the tissue was stored as to its use, but tissue is always useful to us. So if you'd like to contact me, I would be happy to work with you to try and see whether there was enough tissue to save to share with our biobank, and I could coordinate shipment of that tissue to us. But so just contact me and we can talk about it.
Great. And
if I could jump in for a second, Sandy. So there are a lot of hospitals which are sending tissue out for actually a form of testing for the patient, which is a little different than banking. There's a lot of genetic testing being done in cancer today. Groups like Foundation One, or companies like Foundation One, and there are others, Guardant is another, are receiving tissue to do specific genetic analysis. So that is less likely to be something that would be accessible. So it really depends on where the tissue was sent and what it was being sent for.
Thank you, Mark. Since we have Patty, I'm going to give her the second question. How does a patient ask their doctor about redirecting or directing their tissue to us? Is it a difficult conversation to have? Will they be upset with it?
That's a great question. No, I think in general, most surgeons are very willing to help. I would suggest that you just bring it up to your surgeon that you're considering doing it, and sort of let me do the talking. We certainly have ways of convincing hospitals that this is the best thing to do. A reminder is that this tissue really does belong to you, but once it's in the hands of the hospital, like after the surgery, it then belongs to the hospital. So at the time of surgery, there's generally language that I use with any resistance that we come up against, and we can usually convince the hospital, it's not usually the surgeon, but the hospital, that it is the right thing to do because it's the wish of the patient at the time of surgery. So while it's great to mention to your surgeon that this is something you're interested in doing, I think it's even better for me to then follow up with that and just talk with them more thoroughly about it. Hopefully that answers your question.
Thank you, Patty. Thank you. The next one I have for John. Has there been any talk about maybe having Zoom meetings or Google meetings with only fibro patients like we did last year on last year's Vermont trip?
Yeah, and I think I was starting to mention that a minute ago, that we do have 3 more Zoom meetings coming up that basically allows patients who are 18 and over to get together in a Zoom context and, bring a drink, bring your cheese and crackers and meet. And I think Sandy's going to be directing that. We have one for the younger patients, and then we again have one coming up for caregivers. So look on our website in terms of the date for that, but I know the next one is next Thursday. And we believe that this is so important. The other part that's important is with Chan Zuckerberg. It's the same as we've been all along, all of you. It's so important to share with each other, right? It's so important for people to collaborate. So the more that you can all get together and we can help facilitate that, we're happy to do that because the more we learn from each other, the better. So yes, we do plan to do that. And if anybody wants to get actively involved in that, you can go to our website which says join us. There's a tab there and you can fill it out with your name and address, and we'll contact you and let us know how you want to get involved.
Thanks, John. So the next one is a similar question, and I'm going to toss it to myself. It is, I was wondering if there is going to be a call for patient advocates, some people to lead webinars and chats. And the answer is absolutely yes. The patients and the patient advocates are a critical part of our mission and what we want to do. Now that we have our website that is being, I call, renovated and improved, we're going to be calling out for patient volunteers and advocates to come on board and collaborate with us on many, many things. You guys are going to be driving a lot of our initiatives. So I do appreciate the question. John, I'm not sure if you want to add to that.
No, and again, I would say, one of the key areas here is the more we can actually try to get advocates together with the researchers. And again, I'm going to use that as kind of a theme there. The more they can learn from patients, the more things will be advancing. So there'll be a lot of opportunities in that area. Again, things to come in the next couple of months, which Sandy, Mark, and others are spearheading. So please stay tuned. Look at our Facebook, look at our Twitter, look at the website. There's going to be a lot of information coming out in the next couple of months.
Great. Now we have a couple more so we can end the evening, but keep them coming. If you all have more questions for our panel, please submit them Q&A. We'll stay here to answer them for you. Now, this one is for Mark. Oh, no, I'm sorry. This one is for Marna. It's from a caregiver. It is very hard as a parent. Hold on a minute. It is very hard as a parent of a young patient. How do you deal with everything in taking care of your child? How do you deal with
Oh boy, oh boy. Well, your child, your spouse, your brother. It's hard. This is a tough fight. And I don't know. I think you take it one day at a time. You put one foot in front of the other. I think I talked a lot about that in my mother's story on the website, which is long, but Lynn O'Malley won't let me shorten it. So, but you know, there's things in there, but I think to sum up what I probably said in there, I think everybody does what they can do. You know, everybody has different capabilities, and you just do what you can do. And at the end of every day, you know, you just know that you did everything you could do to help your son or your daughter. You made the calls you had to make. You talked to the doctors. You do what you can do. Not everybody can do what the next guy does, and you just do what you can do for your loved one. And that's all we can expect of anybody. You know, even with the foundation, I sometimes think, I wish I was smarter. I wish I could do more. You know, I can do what I can do. And that's all I can expect of myself, you know, and look where we are. So I think for all of you who are fighting this fight or grieving or whatever, just, you know, just take care of yourselves, take care of your loved ones, and you're all doing a great job. You're all rock stars, and do the best you can. That's all that anybody can ask of us.
Thank you, Marna. Mark, we just had a question come in for you. Oh, and we have another one that I'm going to read, but let's get to the. Oh, I'm going to read this one first. I'm going to read it verbatim. Not a question, just a comment. Our son, Austin, was diagnosed with fibrolamellar cancer in 2012 and died in 2014. Kathy and I thank Marna for all your help and look forward to a cure someday.
Amen.
Thank you all for that. Great.
And now we have a final one for Mark. You touched on this a little bit in your presentation, but are we really, really going to see a cure in our lifetime?
You know, Sandy, that's a great question and a very difficult question. I've been involved in cancer research since I was a high school student in some form or other. And there have been an awful lot of promises made, going back, you know, the war on cancer with Richard Nixon 50 years ago, 70 years ago. It's not 50. Well, no, it's 50, I guess. 70, I wouldn't have been alive. I don't want to overpromise, but I do believe very strongly that there are some features that make fibrolamellar one of the cancers that has a chance, a serious chance, to be treated much more successfully in the relatively near future. And there's some strong reasons for that. Number one, it's a genetically relatively simple cancer and it's pretty homogeneous. And the people who have it are young and strong when they start. And that's a big advantage in this fight. And we've learned an awful lot about the underlying mechanisms so that we now have rational basis to try certain kinds of therapies that weren't possible 10 years ago. And where there's some precedent that that kind of therapy might work. An example, there's a rare leukemia called CML, which was really sort of the first poster child for targeted therapy. Guess what? It has a protein kinase that's a fusion protein. You say the same words about fibrolamellar. And a drug was developed called Gleevec that instantly transformed the life of those patients. Suddenly, instead of seeing patients die rapidly when they got to a crisis stage of that cancer, physicians were seeing patients with the disease literally disappearing. Is that going to happen with fibrolamellar? I wish I had a crystal ball to tell you that. But it's not out of the realm of possibility. And I think that's what we should bear in mind as we try to develop those therapies. Immune therapy is, in some respects, in the same category. You know, there's a chance that this vaccine that's being tested at Hopkins could have that kind of effect. The probability is that it won't, but it's probably going to be a step in the right direction. If we see a strong immune response to that peptide, that's a big step in the right direction. And so I think there are tools in place. I think there are good ideas in place. There are terrific people working on it. They're terrific, courageous patients ready to enter clinical studies and who are ready to help us develop these things. So there are a lot of reasons I feel optimistic, and I don't think that's unrealistic.
Thank you, Mark. We have a couple of questions, a couple more comments that have rolled in. So, and it looks like all of our participants are still with us. So I'd like to maybe go through some of these comments and questions if everyone's okay. This is the comment. We are a new Fibro family as of June. I would love to see more from the Fibro Foundation when it comes to bringing families and fighters together. Besides the Facebook page, family support is very needed. John, I know that we've been working on this a lot. Perhaps you would like to comment back?
Yeah, and again, we support all of this. And again, I would say take these great comments and please go to the website and under the Join Us and put those questions or those, those thoughts in gear. Again, we have added in staff so we can actually respond faster to you along the way, but we understand the need. We understand that it's important. And again, just make sure that you share that so that we can kind of come back to it. But we do understand the need, and again, we'd love to address it.
Yes, we would. There is a similar question, of course, with a patient support group. And they would love to have a fibrolamellar cancer patient support group created. So we have some comments coming in with that. Now, this one is in response, I think, to you, Mark. It's just a comment. I just wanted to add that when I was diagnosed over 15 years ago, there was no talk of even care or a cure. This organization has given me so much hope that we will get a cure. So, yeah. So for everyone interested in the support groups or any information, please reach out to us on the website. And with that, Marna?
Okay, well, thank you all. And thank everybody for coming. This is the first of what we hope are going to be many webinars. And please, for those next week, next Thursday is our second webinar, and it's going to be interactive. I know a lot of you have been asking, can we talk to each other? That's many of the upcoming ones will be interactive, and next week's will be next Thursdays, and it's going to be for 18-year-olds and over. A week beyond that or so, it's going to be for the under 18-year-olds, and a week after that, it's going to be for caregivers. And I do see this. I think that the fact that we do need to continue that gathering that David Birch hosted last year in the Ice House. When you all got together, there were some funny comments coming from that night, David, I remember. But it was turned out to be good, didn't it? And just for patients to get together, and just talk to each other. So I think it's a really good idea. So maybe Sandy can get that going, and a good idea. But anyway, thank you all for tonight. We've learned a lot, and I look forward to seeing all of you next year in Vermont. Okay? Love it. Good night. I'm leaving.
Good night, everybody.
Good night.